Good afternoon, everyone, and welcome to this short webinar. It's going to be a first webinar that we do on this topic of reproducible bioinformatics and the biocompute objects, and we hope to continue and do more of these sessions in the future and incorporate them into some of our training programs. So I briefly wanted to, first of all, talk about why we are doing this and where this is going. So as you can see, we have a bunch of different logos here. So we wanted to start introducing the biocompute objects in the context of the bioinformatics training that we are conducting with Louisiana Biomedical Research Network and some of our other partners and universities where we have these programs. So we wanted to, first of all, introduce Jonathan Keeney, who's going to be speaking today from George Washington University and part of the biocompute organization. And I'll let him talk a little bit more about that. But we also wanted to use this opportunity to kind of explore the Precision FDA BioCompute Object App-a-thon. And I'm sure that Jonathan is going to be mentioning that as well. But recently we learned about this and we wanted to see whether some of our projects that are in development, some of the research projects that have been initiated between the faculty and the students might be a good fit for this challenge. Unfortunately, the challenge has only 15 days remaining, but as we will see today, this is not as complicated as it might seem. So maybe we will find a good fit there. And so to introduce everybody to this concept and to the challenge itself, we're going to do the webinar today, and we're also going to try and do another one, maybe at an earlier time, so some of the international participants can join as well. We will also be recording this session and then making it available for anyone that's a part of our projects. So now I just wanted to introduce Jonathan Keeney. Jonathan Keeney is part of the executive steering committee for the BioCompute, and he's also an assistant research professor at the George Washington University. So he's going to be speaking about the BioCompute project and giving us a little hands-on tutorial. Some of you might want to participate in trying to replicate this yourselves, but of course there are some registration aspects that we might not be able to complete, but at least you'll get a glimpse of what this is and realize how important this might be to make our bioinformatics projects more reproducible, and ultimately in the future leading to the FDA and regulation being a big part of this as well. So I'll turn it over to Jonathan. Jonathan, if you can try and unmute yourself so we can check that everything's working.
Okay, well, thank you very much for the introduction. We can probably do a little, probably not too much hands-on. I think, as Lilia mentioned, there is kind of a sign-up steps, so we can try to get going on that as much as possible. And I'm happy to stay on and help out anyone who does want to try it out. But I'll at least show you to where that is and show you how you can use some of the tools that we've developed to try and work with the BioCompute standard. So I'll talk about this in a kind of a very high level, starting from a very high level and kind of narrow it down, assuming that nobody knows anything about it. So just from a very high level, the sort of punchline with BioCompute is that it's a way to collect and communicate all of the details of a bioinformatics workflow. So if you were to go get your genome sequenced, you would get maybe hundreds of millions of raw reads. These are all ATCG and so forth. And those are meaningless to a human. So in order to be able to turn that into anything meaningful like ancestry or cancer or microbiome, disease correlation, things like that, there's a certain number of data transformation steps that have to happen, sometimes an extraordinary number. The number of steps in pharma can sometimes exceed 100. And right now, it's kind of a black box because this is very much an exploratory phase, and that's wonderful, and we're all learning a lot. But when it comes to having to communicate that from one place to another for a variety of reasons, it becomes very difficult. And so oftentimes what happens is a bioinformatician and their team might work on the data for maybe 6 to 8 months, kind of trying things out, seeing what works, trying other things if it doesn't. And by the end of all that, the original architect of the pipeline understands what happened, and the entire pipeline is very clear to them, but it's not clear to anyone else, and it's sometimes not reproducible. And so this is very similar to what you might see in a wet lab experiment. There are certain inputs that need to be reported with the experiment in order to make sense of it. So you couldn't just be given a fluorescent microscope slide, for example, and necessarily know all of the details about it. You need things like strain of the animal, what's the tissue type that you're looking at, some of the more granular details like the protein lot number if you're doing a fluorescence in situ experiment, things like that, that are not, you can't intuit those kinds of details if you want to be able to repeat the experiment. And bioinformatics is no different. There's a number of inputs and parameters that are needed to be able to reproduce, or in most cases even understand a bioinformatics pipeline. And so this has been a real challenge, not just for folks that are new to bioinformatics, but even for experienced bioinformaticians, because it's oftentimes not clear what all needs to be communicated. And so one of the biggest places it's been felt is in academic publishing. There's been this replication crisis going on in terms of being able to reproduce experiments. And I think many of us are aware of that, but another one of the major places where it's been very problematic is in submissions to the FDA. So oftentimes what happens in submissions to the FDA is an industry sponsor will submit some data, and that if that includes any sort of bioinformatic pipeline, the FDA will review it. Usually what happens is they'll have to kick it back to the industry sponsor with need for clarification or issues or questions or something along those lines. Excuse me. And the industry sponsor will then have to figure out what it was that the FDA didn't understand, because this is not a situation where you can sit down next to someone at a computer and just sort of walk them through what you did. Every time this data changes hands, it goes through lots of attorneys. And so this can add months and months of time and millions of dollars. It was at one point estimated maybe $6 million for one of these. The industry sponsor then tries to address those things and resubmit. In fact, there's actually sort of a funny anecdote about this. At one point, an industry sponsor submitted this to the FDA in question. In this particular case, reached out and said, I have a question about this bioinformatic pipeline. What did you do here? Or what did this mean? And they said, we don't know. The post student or a postdoc leaves. Needless to say, that application was not approved. So the industry sponsor will then resubmit, and sometimes they can go around this loop many, many times, adding even more time and even more cost to it before the FDA finally just has enough to be able to make a decision. And this is a really, really big problem, because the FDA recognizes the power in precision medicine and the fact that bioinformatics analyses, in particular NGS, or HTS, as we're saying, high throughput sequencing data can be for precision medicine. It's really helping to power a lot of this. But when it comes to actually getting approval for these things, it's very problematic because it's taking up the time and it's taking up the money, but it's in no way improving the probability of getting an approval decision. And so essentially all it's doing is increasing the risk for one of these. And that's a really big problem. So it's a big problem that kind of needs to be addressed. And the way that it's traditionally been handled is each individual reviewer will put out their own document that sort of explains, if you're planning to submit NGS data to us, here's what I recommend you do. These are not standardized documents. As you can see here, it's not an official guidance document. This is just the opinion of one reviewer, and it's based on their experience. So it's very much trial and error, sort of how they've figured out from these situations where it didn't work, what sorts of things you can do. And so it's very piecemeal, and it's not standardized across the FDA. It's not even standardized across centers at the FDA. It's very much sort of ad hoc, and it makes it very difficult to be able to predict what the FDA wants in terms of this data. And it's been a challenge, quite frankly, because it's new enough that I think the FDA has not been able to say what they want from the data. So there's been a very big drive to try and address that. And so the FDA convened a special session of the Genomics Working Group. And this is a working group at the FDA that has broad expertise in genomics across lots of different fields and across all the different centers. So they have representation FDA-wide. And so in 2013, they convened a special session to address this, and that working group came up with this bulleted list for what any sort of solution should have. And these are the 4 bullets. It should be human readable, sort of like a GenBank sequence record, if you're familiar with that. It's just a name value pair, or key value pair, as they say. It should be computer readable, meaning any data that's in there should be structured and adhere to that structure in some very predictable way so that you can work with that data if you need to. It should contain enough information to understand the computational pipelines, interpret that information, maintain records, and reproduce experiments. That's really the core of the entire concept. And then finally, there should be some way to make sure that the information has not been altered. So once you build a record in whatever way this is, it has to be immutable. If you do anything to change it, then it's no longer the same thing. So the solution to that is what we now call BioCompute. So this began as a collaboration between George Washington University and the FDA. It's a standard, well, it's a forthcoming standard, rather, for communicating genomic analysis workflows. And this should really say bioinformatic workflows, in fact. The heart and soul was born with genomic analysis workflows, but it's really very well adapted to any bioinformatic pipeline at this point. We've tried quite a few others, including building databases, for example. It acts like an envelope for an entire pipeline, which means that it can incorporate several other standards that include CWL or WIDL if you're using either of those. So that would be good for portability of execution, for example, particularly if you're using command line. FHIR Genomics is another example, so that's fast healthcare interoperability resources. That's good if you're working with patient data, for example, and you are building apps on a mobile phone or a tablet or something like that. So a much more kind of clinical and applied side. So it works very well with all these other standards. SeqC2 is another example, GA4GH. So it was built with the FDA, both with input from FDA personnel in terms of what their needs are and with FDA funding initially. We recently just received a grant to start training some more of the FDA folks, as a matter of fact, and gaining even more use case scenarios. And we have a publication that is, well, I should say a manuscript in preparation right now that is kind of walking through a mock submission using the BioCompute process. It was written to be human and machine readable, so it's written in JSON, which is JavaScript object notation, and I'll show you an example of what that looks like in case you're not familiar with that. But it's a very simple, easy to use language. It's not a programming language per se, it's just a way of structuring data. It's categorized by domains at the top level, so these are sort of ways to conceptually break down a workflow in ways that make a lot of sense. So if you were interested in the metadata, there's a place to look for that. If you're interested in the parameters that were used to actually run the steps, there's a place for that, and so forth. And I'll show you an example of these things. It adheres to the FAIR principle, so it's findable, accessible, interoperable, and reusable. Each of the letters in that acronym has a very specific meaning that a pretty large international community has agreed upon. And we've got a few representatives that have been part of the BioCompute consortium that have been very strong advocates for that. So BioCompute does conform to the FAIR principles. It's very adaptable. It's got an execution domain in it as well. Sorry, not an execution domain, an extension domain. And so the extension domain is a user-defined domain that allows the user to tie it to any other schema that they'd like or any other resource. So that makes it very adaptable. It preserves data provenance. There's actually an entire domain, the provenance domain, that lets you track who transformed that data. And it's also got an embargo field and a sort of a time valid on it. So sort of like an expiration date on it if you need to do either of those things as well. The provenance domain in particular has been very appealing for certain groups. One startup, as a matter of fact, based in Boston, who is trying to kind of transform the way that individuals will contribute their genome. So instead of getting your genetic data from a 23andMe or an Ancestry.com or something like that and checking the box that says, yes, I would like to participate in research, at which point they take that data essentially in perpetuity and sell it when and however they'd like. The goal of this particular startup is to create an online marketplace where individuals retain rights over their data. And so if say a biotech company gets onto the marketplace and searches for white males between the ages of X and Y, and my profile shows up, they can make an offer to use my genetic data in their clinical trials, and I have the option to say yes or no. And so this is kind of nice because it keeps the incentive for individuals to continue volunteering their data, their personal genetic data for that reason, but it does so without stripping them of the rights of that data in the future. So I think it's really exciting. And they're using BioCompute as a framework for their entire marketplace because primarily of that provenance domain. So you always have a clear record of what was done with that data and by whom and when. So it keeps everything very transparent. And finally, it has unique IDs for versioning. And so this was how the problem of being immutable was solved. And so the unique IDs, there's a registry where you can register your domain, and then your own organization can kind of determine what the value for that unique ID is. But if anything changes, then the unique ID has to change. So we have a registry that lets you do that, and an aggregator that kind of compares them. So once you build a record, which is called a BioCompute object, or a BCO, so it's a specific instance of a pipeline written in BioCompute, then it gets its own unique ID, and that's it. So if you change anything, then you then have to version the unique ID. And so a good analogy for this that I like to give, because there has been a little bit of confusion over what exactly the standard is standardizing. A good analogy for this is Wi-Fi, which is wireless internet. That's more formally known as the 802.11 standard because they went through the IEEE standardization process, Institute for Electrical and Electronic Engineers. And so Wi-Fi is a good analogy for this because Wi-Fi does not standardize whatever product you're making. You know, if you make a Mac or a PC or a printer or a toaster or whatever you make, all you have to do is configure that device to use, or rather to be compliant with the 802.11 standard. And they don't try to standardize the platform that you use. It could be Mac or Windows or FaceTime or whatever. They don't standardize the data that you're using or the apps that you're using on the computer. You know, the only things they're standardizing is the way that that information is collected and reported between two entities. And so that's a very good analogy for BioCompute. You can use any platform that you want. You can use any sequencer. You can use any bioinformatics tools or pipeline that you like. You can develop your own pipeline. That's all fine. This is just a way of collecting and communicating that information back and forth between two entities. And so this could be the FDA and a pharma company, for example, instead of a router and a Mac. Okay, so that's a brief introduction to it. So this is sort of a crushed down snapshot of what a BCO would look like. So it's color-coded just for clarity here. So the top section is all metadata. It's all written in JSON, which, as I said, is just key value pairs. So ID, what's the ID, name, what's the name. There's no logic in JSON. It's not, there's no if then else or for loops or anything like that. It's, as I said, it's really just a way of organizing the data. It includes a usability domain, which is just in plain language, what did I do and what was I hoping to find in this, sort of an executive summary of the entire pipeline. This is also where you could include details about the pipeline itself, since this is metadata. So for example, if you ran a pipeline for one specific purpose, but you just wanted to include a note in there that says, if you wanted to use this for this sort of similar related purpose, you might consider changing step 4 into X, Y, and Z. This would be the place to do that. There's also a description domain, which includes keywords and cross-references if necessary, and pipeline steps. I'll show you an example of those. The execution domain is the platform that it was run on, and that has to include things like any drivers and scripts and other prerequisites that are necessary to execute that, anything necessary for deployment, essentially, along with the versions and ways to access them. Parametric domain is a record of all the parameters that went in, and this is specifically just the parameters that were changed. So anything that's not default. So for example, if you're running a pipeline at the command line, a lot of these command line tools have default options built in. So if you change any of those options, this is where you would record that. But anything that you don't change does not need to be recorded. And so each tool will have its own specific set of parameters that can be changed, and a different number of them. And many of them, therefore, will not have anything that's changed. After that is the I/O domain, which is the input-output list. And so this is just a list of files. Rather, it's a list of where you can access those files in the form of a URI. We don't actually keep any of the data on board with a BCO. These are just references or pointers to where those files live. So the final file size of a BCO is on the order of kilobytes. But we still do include a record of where those files are. And then finally, the error domain. And so this would be where you keep information like false discovery rate, other metrics that you might have benchmarked your pipeline for that you can kind of use to sort of demarcate the bounds of the pipeline. So what's the minimum read quality that your pipeline is able to work with? What's the minimum read depth, for example? And so the I/O domain and the error domain are collectively what we're calling the verification kit, which means given some input file or files, and given this error domain, is the output domain valid? And so that is sort of a quick control test that can be run to make sure that anything that's being submitted along with this pipeline actually is valid. It should, in fact, be detectable by this pipeline. So that's essentially what the pipeline is. Right now, we're currently working with 1.3, version 1.3, and this is a link to that. This is all on GitHub. The 1.4 version is very close. This is going through IEEE standardization right now, and it's already passed the major vote, and it's already gone through commenting. So it's passed the ballot, it's passed the commenting, it's currently in comment resolution and very nearly complete. So I expect this to be submitted to the formal IEEE voting process at the end of this year. And so probably early 2020, I would expect 1.4 to be live and to hopefully be an IEEE certified standard. So, so let's see, are you able to see this screen now, the browser screen?
No, we actually still see your presentation.
Okay, let me switch over to this right here.
Yeah, now we can see your screen.
Okay, great. So this is the 1.4 is now being kept on GitLab. There's been some changes with this one, and so we've got a different kind of an organization for each of these domains here. So each top-level domain has its own specification here that's referenced by the main, which is biocomputeobject.json currently. And so one of the big changes is that we are building the entire thing on the JSON schema. So we inherit all of those automatically. And one of the sort of big upgrades, I guess, that we get from that is the ability to include the required and optional, and that's not present in the 1.3. And so in the main file, you can see all these domains that I talked about, the top level domains that are sort of the conceptual breakdown are required. And then in addition to that, there's 3 that are required: the BCO ID, which is the unique ID of the BCO, the spec version, which in this case will be 1.4, and then a checksum, which we're using SHA 2564. So then each of these different domains has separate dependencies therein. So here's the description domain, and you can scroll down and see this is where the cross-references, the space where the cross-references are, the properties, access time, et cetera. This is the platform, as I mentioned. All of pipeline steps would then come under here, along with the descriptions and the access times and the URI. So this is the current version, the 1.4, that I think is very close, and so we can run conformance checking against these schemas. And so I encourage you to go take a look at these if you have any interest. As I said, we're still currently on the 1.3, which is let me switch back to this here. Okay, we are still currently on the 1.3, which is still functional, and in fact is the one that, at least at the moment, still has a manuscript being written about. But I expect 1.4 to be coming out soon, and so there's a couple of groups that have already started taking a look at that to see how they can work that into their workflows. There we go. Yeah, so just for a very quick example, we can just build a quick kind of pipeline and step through what it would look like. So, for example, if you're a researcher and you've got a cohort of individuals that have HCV1, hepatitis C, and they're all in this drug lidiposphere, and there's a subgroup, a subpopulation there that's drug resistant. And you'd like to explore that and see if maybe the virus has acquired some sort of mutation. So that's the working hypothesis. And so you're able to isolate some of the virus from this drug-resistant cohort and sequence it. And so that's what you've got to start with. And so we can just build a simple two-step pipeline to investigate that. In this case, I'm just showing you an example on the Hive platform. The Hive platform is the major bioinformatics platform that the FDA uses. In fact, it's the only one aside from command line that they have available inside the firewall. We happen to have a clone of that here at GW, so we're able to build on our own tools. So this example is Hive, but as I said earlier, it's platform agnostic. You can do it on any platform you want. This particular tool that we're using is Hexagon Aligner, and it is pretty straightforward. You input parameters, you can watch the progress, and then look at the results. And so that's the screen we're on now. See that everything worked out pretty well. There's a pie chart breakdown of everything, so that's fine. Next step would be to look for variants with a goal of trying to find any variants that are consistent only in these drug-resistant individuals, but which are not present in the reference, because those would be a strong candidate for being causal variants. And so this tool is called the Heptagon, which uses a VCF or a variant call file, and very similarly entered all the parameters, watched the progress, seeing the results. And so in this particular case, they found what they were looking for, and so we're zoomed in on this window where you can see this high degree of variability here, or variance rather. And so this is a pretty interesting case of a potential precision medicine usage. And so any individual that has HCV1, if they get the virus sequence and they have this mutation, might not be a very good candidate for lidiposphere. And so there would, of course, need to be some follow-up to investigate that. But this is a nice example of exactly that. So building BCO is sort of like building a recipe card if somebody else wanted to do this. So if you went to somebody's house and had this really wonderful salmon and you wanted to repeat it, and you went home and repeated it, and it was nothing like what you tried, and they said, well, where did you get the salmon from, and where did you get your ingredients from, and where did you get the spices from, and things like that. This is an example that one of the FDA folks used, and I liked it so much I'm trying to repeat it, and hopefully only mildly butchering it, but I think it does a really good job of kind of getting to why, sort of at the top level, it may not seem as necessary because you just sort of write down the steps, but in reality, there's a lot of granularity that goes into each of these steps. And so what you would do in the case of Hive is record all of these scripts and drivers and prerequisites for each of the steps along with any relevant parameters, and include all of that along with the execution domain, which is the platform that it was run on. And so in this case, that was Hive. So you'd have a full step in both the parametric domain and description domain for the hexagon and then for the heptagon. You would then add in pointers to the input and output domains and have timestamps on all of these things, and then layer on any necessary usability domain and keywords and so forth. And then if necessary, also an extension domain here that describes, in this case, a GitHub URI. So if you want to make elements of this or the entire pipeline available on GitHub, you can do that as well. And so then you've got this BCO that can be shared very easily with other researchers or with a publication if you want to publish it, or with the FDA if you want to submit it as part of a clinical trial. And so that's a simple two-step pipeline. You can imagine this could be used with any number of steps. In fact, the more steps there are, the more useful the BCO actually is. And if you were to actually open up one of these BCOs and look inside, what you would see is still relatively easy to read, even if you don't have a text parser. So this is the usability domain. This is just in plain English. So wanted to identify baseline single nucleotide polymorphisms, et cetera. So just the entire experimental procedure that I explained earlier. The pipeline steps then start to lay out each of those steps. So the first one was the Hive hexagon. The description of that is just to align alignment of reads to a set of references, which version was used, an array of prerequisites. So in this case, there was multiple prerequisites. So they wanted to use the hepatitis C virus genotype 1 as a reference to do the alignment. So that comes in as a prerequisite. There's a URI for where they address that and an access time. In this particular case, they felt that they wanted to use the HCV type 1b complete genome as well. So that also gets listed here. Any of the prerequisites that were necessary for any of the steps would then get listed in this section here, the pipeline steps in the description domain. Execution domain then has all that information about the platform. So again, this one was called Hive. There's information about the version, the prerequisites here, again, along with all of the URIs and access times and so forth. The parametric domain, so in this case, there was only 3 parameters that needed to be included for hexagon, seed, minimum match length, diversions, and then 2 for the Hive heptagon. This is also where you would include any command line arguments that were necessary to invoke the command. So -H or -A or use strict or whatever it is that you use, those kinds of things would go here. And then the IO domain at the very end. So we've already started integrating this into a few different platforms. It's on 7bridges and the Cancer Genomics Cloud, or CGC. They have that integrated, so there's a record biocompute button that lets you export all of the stuff that can be exported relatively easily. You still have to fill out some of the metadata, like the usability domain and so forth, and who you are and things like that. But you can automate most of it since it's in a JSON, which is sort of the lingua franca of all programming languages. We've also got it into Hive. There's a record biocompute button there. We are currently working on a grant to get it integrated into Galaxy, so there will be a nice record biocompute button built into that. Sort of doing it in 2 approaches. There's a toolshed version and one that's integrated into the main code. And then hopefully soon get one on DNA Nexus as well, DNA Nexus and Precision FDA, which is powered by DNA Nexus. So these are platforms that already have the ability to build biocompute objects on them already, which makes it very easy. So once you're done, you just export the whole thing and then add in any relevant details that you want. Some of them are also currently working on readability, so you can import a biocompute object and have it have those details ready to go, and then execute portability would be our next step, kind of looking down the road. So you could not only import a BCO, but then have the ability to execute that entire BCO right off the bat. And so we're working with groups like the Association of Pathology Chairs, for example. One of their sort of subgroups called the GOAL Consortium is interested in standardizing the way that cancer is diagnosed using these NGS platforms. And so every cancer, of course, is going to have its own set of needs, but they'll all, in theory, have a core set that they're interested in. So maybe RAS mutations, for example, would be on that core set. And they've got, in fact, I think a few hundred genes that are part of that core set. And so we're working with them to try and build a BCO record of every iteration that they're building to try out, and then have a BCO record of the final version that can be shared with all the pathology labs around the United States. So if you're not working on one of those platforms that has it, or if you're just working at the command line, we also have a form-based editor. And so this is a way of trying to make it easier to build a BCO without actually having to code the JSON yourself. We do actually preserve the ability to work with JSON. So there's this Edit JSON button, so you can click that and open up the JSON and either for the entire thing or by domain. And so the URL for that is here. It's www.biocomputeobject.org/BCO_editor_TST. And so this is what you can go to today if you would like. You'll need to go to register, and I can activate your account as soon as I see any of those come in. And so you can take your favorite pipeline, or you can take a publication if you'd like, and start to build a BCO out of it. You can save your changes here as you're working on it, and then come back to it later if you'd like. The only thing I will caution you on is don't hit the back button or try to close the tab or anything without saving it, because then you'll lose all of your work. But you can save the changes and then come back and edit it later, or download it if you'd like to do whatever you'd like with. So these are some of the groups that we've worked with. We've worked with a number of groups in both the public and private sectors and in academia. I haven't put everyone up here, but I tried to put some of the bigger names, and I won't go through all these. I will mention really quickly, though, Google, because we began working with them. So they've agreed to use the BioCompute standard to host all of the STRIDES data. Or I should say, host all the data as part of the STRIDES program. So that's the NIH program to make all publicly funded data available. And so they're doing that through both Google Cloud and through Amazon Web Services. And so Google has agreed to make all of this data BioCompute accessible, BioCompute compliant. We also recently had a workshop at the FDA. And so at this workshop, we had a discussion panel with the Genomics Working Group that originally came up with the idea to do something along these lines. And so they talked about how workflow communication is currently being handled at the FDA and what some of the challenges are with that. And so we had FDA reviewers, FDA researchers, some academia folks, some folks from private sector, and one patient advocacy representative on that discussion panel. And that, I think, really was the highlight of the entire workshop. It was really good. And there are recordings of that as well. There were use case examples. I can go over just a couple of those very quickly. There were demos and hands-on, mostly for the things that I showed you, the 7 bridges and the BioCompute Editor. So demos and hands-on for those things. And then finally, a BioCompute challenge on Precision FDA, which Elia mentioned, and which hopefully some folks here can contribute to. So at the end of this, we can as I said, you can go to the BioCompute Editor and get started working on it. And if there's a pipeline that you would like to record as a BCO, it would be really cool if you submitted it to the BioCompute Challenge on Precision FDA. That'll get reviewed and possibly even have a chance to become part of a publication. We're kind of exploring the idea of doing that. And any publication, of course, would have to go through the normal peer review steps. But what we'd like to do is try and get the top performers to participate on a manuscript around this. So that's what we would like to do. So if you have any interest in using this, I think that's probably the fastest way, is to just jump in and start building a BioCompute object and then submit it to the Precision FDA BioCompute Challenge. I will say anything that you do submit needs to be in the public domain, or you need to be okay with it being visible by the public, because this is a public website. So please don't put anything up there that is patent protected or that you're not able to show to the public for any reason. So some of the use case examples that were presented at that workshop are tuberculosis detection. So TB is a top infectious killer in the world, and the WHO has adopted the Reseq TB pipeline, which is a great pipeline for this, but it still has some of its own challenges, which involves things like lineage identification, prediction of antibiotic disease, recurrence, and so forth. And so they're exploring BioCompute to try and help navigate some of these challenges, and in particular, the provenance domain seemed to be very helpful. So they gave a talk about using BioCompute for their efforts. We had a test submission. This one was with HCV1A. Coincidentally, this was not the example I showed you earlier, but this was the mock submission where we worked with a bioinformatics company and with an FDA reviewer, and with guidance from the FDA reviewer, came up with a mock data set. We gave it to the bioinformatics company, who ran it through their actual pipeline. And we used that as a benchmarking. Since we came up with the data, we know what truth is, so to speak. And they ran it through their pipeline and generated a report. And then we were able to use that as the verification kit and run an actual mock using a larger synthetic data set to submit to this FDA reviewer, who then wrote up a full report that detailed his understanding of it based on the BioCompute and the BioCompute alone, or the BCO, I should say, the BioCompute object and the BCO alone. And then at the same time also tried to generate his own BCO using that data. And so that was a really cool, really exciting one. And so that's in preparation right now. And then the last one is Emblema, which is the startup that I mentioned earlier that's building an online marketplace for buying and selling personal genome data. We are currently going through IEEE, as I mentioned earlier, our working group. And so IEEE is the Institute for Electrical and Electronics Engineers. It's a very large standardization body. Our sponsor society is the EMB, the Engineering in Medicine and Biology. And our working group is P2791, if you have any interest in watching the progress on this. We're hoping to have the final one submitted in time for the last voting of, or the last deadline of the year, which I think is December 3rd. So we've still got a couple of months before we need to get that in. But I think we're already ahead of the game because I think it's very nearly ready. So we've already gone through, it's already passed the ballot and it's already gone through public review. And so now, as I said, it's in comment review, comment resolution group, or CRG. And so it's very nearly finished with that as well. So this will be our 1.4 version, which would be the first to be passed by IEEE. At the same time, we're also hoping to go through the International Standards Organization, or ISO. That would give us additional stability. It would give us additional reach, I think. It's an even larger society than IEEE. And so IEEE and ISO actually have a joint agreement in place whereby anything that the IEEE accepts and publishes as their standard, the ISO has the ability to publish it sort of as a joint standard. And so we've been exploring that with the SC32WG2 working group at ISO, which is convened by Denise Warzel at NCI. And so she's been an observer in our comment resolution group at IEEE as well. So she's keeping tabs on the progress there. We've also recently launched a public-private partnership. This is a way of sort of bringing in anyone in public or private or anywhere that has any interest in weighing in on the biocompute standard and the way that it's built and the way that it's used and so forth. And so we recently signed with CBER at the FDA, the Center for Biologics, and what is it? Center for Biologics, and I'm blanking on the name right now. Evaluation and Review. So they've signed a public-private partnership. We will start bringing on other partners as well very soon now, once the Executive Steering Committee finalizes a plan for that. And so the primary goal of this public-private partnership is very much complementary to all the standardization efforts at IEEE and ISO, which is to develop a community of stakeholders with interests in this area to facilitate the development of tools and facilities for use in coordination with biocompute, and to facilitate the portability of pipelines for execution. And so it's guided by an executive steering committee chaired by Raja Mazumdar, and I'm acting as the managing director. These are the other members of the committee. And then there's a technical steering committee, which is chaired by Hadley King, also at GW, with some incredibly talented people from Internet 2 and NCI and FDA and Galaxy and Common Workflow Language and several others. And this is really, I think, the heart and soul of biocompute. So these are the guys that really, really understand the technical aspects of it and the need for it and how it can be built and used. And so they'll help actually building tools and help out with some guidance in terms of implementing it as well. So, right, so this is the editor I was talking about. So if you have any interest, please go to biocomputeobject.org /BCO_editor_test and fill out this registration. And I will activate you as soon as I get that notification. And then you can start to build BCO objects on there as soon as you're ready. And then once you're done with it, download it. And as I said, if you can go to BioCompute App-a-thon, which is the BCO challenge on Precision FDA, there's actually two tracks here. There's a beginner track and an advanced track. So if you are a little bit more on the technical side, we also have this advanced side, which is to submit a conformance tool and a writer tool. And so this conformance tool will check any BCO against the specification. And the writer tool is something that will have the ability.
Sorry, Jonathan, can you maybe paste the link in chat right here, or maybe go back to that previous slide real quick?
Yeah, so you're talking about the App-a-thon site?
No, I'm talking about the BioCompute where you,
Oh,
yeah, that one.
yeah, there you go.
So anyway, you could just paste that in chat for everyone, or?
Oh, yeah, sure. Let me get out of there. Where is, here we go. So let's see, where is, how do we get to chat? Okay, let me pause sharing.
Okay, here, I can do it for you if you want. Okay, it's in there.
Oh, you already got it? Okay, sorry about that. Okay. All right, so are you seeing this web page now?
Yeah, BioCompute Editor 1.4, and you have the login or the registration, right? So you mentioned that people will submit their name, and then once they click on register, you are the one that gets that?
Yep, so we'll get it here, and I can, I mean, I'll be here at the computer, so I can start activating people immediately. And so all you'll do is go to Create Object in the top right there, and then I'll pop up this example for you that you can work from if you'd like. And then it'll take you directly to this editor here. And so this is where you can start inputting all of your information if you'd like. If you want to include a license, you can include that here. And then for each portion of this, you can add an item if you need to. So for example, in parametric domain, if you've got item 1 and you fill this out and you want to add another one, you can just add that right there, and there's item 2, or delete it. And again, you can always go to edit JSON and just edit that directly if you'd like. Now when you're done, just come up, you can come up and just save the entire thing, and then download it if you'd like. So here's save changes. And then back at the list, you can download the entire thing as a JSON, or you can always just go to edit JSON here and copy the entire thing and paste it as well. So there's 2 different ways to do it. So that is the basics for that. How do I get back to, oops. Yeah, so, and then the very last link that I'll share is the App-a-thon site. So this is precision.fda.gov/challenges/7. And this is part of the Office of the Chief Scientist, Office of Health Informatics under Elaine Johansson. So that is BioCompute and the Precision FDA Challenge in a nutshell. And I'm happy to hang out here and kind of help anyone who does want to walk through that. But for now, I'm also happy to take questions.
Yeah, thank you, Jonathan. And I just want to add to that what we want to do is try and work through some of the projects that are ongoing that we've had in different programs, and hopefully work through some of these examples that we have and try to submit them. So if you are a little bit lost as to how to use this, you know, we'll reach out with another time that we could schedule one of these webinars and kind of do this together. And of course, those of you that are already working on, you know, different bioinformatics pipelines and projects, I think there's a way to register yourself. Again, we have that link on the chat. And right now, if there are any questions, we can try and answer those. I'm sure Jonathan can have any input in, you know, whatever you're working on.
Hi, Jonathan. This is Pratim. Can I ask you 2 questions I have?
Yeah, sure. Go ahead.
Yeah. So one of the thing was that, you know, you talked about the JSON objects, which are metadata exchange format. So you already have defined objects in the JSON schema. Is it possible to create new objects which may be useful for a specific project or something like that?
Yeah, using fields that are not already defined. Is that the question?
Yes, something like that, exactly on that line.
Yep. So there is an extension domain, and that's, as I said, that's sort of like a user-defined field. So we kind of leave that one open. It doesn't have any really any rigid kind of guidance on that one. So what we have kind of recommended as a best practice is that you build your own schema of any kind, and then include a URI to that in the extension domain. So that's the way that we can validate against it most easily, is if there's just a URI in that field.
Okay, thank you very much. And one other question I had, let's say, I have, you showed us two examples specifically on this HCV1, and there was another one, cancer, I presume, I forgot. I mean, let's say there are two therapeutic areas. Let's say one is a cancer, one is an infection. So is it possible that, both of the same, the same BCO objects are kind of used for both the therapeutic areas, or both the, both the same problem sharing common BCOs? Is it kind of thing that might occur?
Yeah, well, so I think that is kind of the goal with the standardization of cancers. The diagnosis of human cancers. That's sort of the goal. Sharing across different areas is, yeah, it's certainly possible. I think it's less likely. When you do sequencing of viruses, for example, versus humans, you might have a different seed size and minimum match length and things like that. So some of the details there, kind of, my intuition is that it's unlikely that those would be shared across regions that far. But we do certainly expect that some of these are going to be very much standardized. And as we begin to figure out which pipelines work the best, there will be sort of a natural demand for those ones. And there will be sort of a natural demand for authenticity of those pipelines. So there's got to be some way to get it and have a lot of confidence that what you've gotten is authentic and has not been tampered with. And so BioCompute kind of has a very natural way to help facilitate that. So you can have authenticity kind of built in there, and we have an aggregator that lets you compare a hash against, like an SHA-256, for example. So yeah, we are very much expecting that there are going to be some instances where there's sort of a high demand for a BCO that's used in more than one instance.
I mean, overlap between the two things.
Overlap between, like, let's say, infectious disease and cancer?
Yeah.
Yeah, so again, I don't know. I guess it would just kind of come down to the specifics. I certainly don't want to say that it's impossible that something like that would happen. My limited experience looking at BCOs in those two different realms, as I say, my intuition is that the needs of each of those pipelines tend to be different, but I can certainly imagine that it could happen, that you'd have a BCO that, for whatever reason, is very useful in both of those areas.
Yeah, thank you very much for your answer.
Sure.
Philip Kilgore from LSU Shreveport here. I was just wondering on the topic of integrity, because this is, a compelling interest that you mentioned as part of this. What made you choose a straight hash over, let's say, a digital signature?
So that actually came up during the discussions that we had back during the working group. And unfortunately, the security hash is not really my area of expertise, but there was a couple of folks who have worked on projects like this, and they felt reasonably confident that using a hash was a pretty good way to go with it. It has not formally passed the finish line yet. So if you felt for some reason there's a really strong reason that we shouldn't use that, then I would certainly welcome you to submit that issue on GitLab and suggest something alternatively. Do you think that a digital signature would be stronger than a hash?
Yeah, and part of the reason is with just a straight hash code, somebody can construct a rainbow table for it. And so the rainbow table is basically like a database of plain text to the hash codes.
Mm-hmm.
Now, if you end up with 256 bits, right now that's actually quite large. It might be infeasible to construct a rainbow table of that size. It's quite a large amount of data now. But in the future, it may turn out to be the case that a particular SHA 256, I'm not saying that there are weaknesses now, but there may be weaknesses discovered in the future. And the nice thing about the digital signature is that not only do you get the integrity out, but you also get the non-repudiation, so that you can say that this entity was actually responsible for providing this pipeline. That's something worth considering.
Yeah. Yeah, well, I know that there was news that I guess it was Google has released or not released, but developed the first quantum computer, right? So maybe it's time to start thinking about things that are not hash-based, right? The strings like that, right? So that's a fair point. We do have the initial, so there's a registry that lets you kind of establish a domain. So for example, we would be GWU, which would kind of prepend our unique ID. FDA could come up with theirs. So you can go register a domain just like as if you were GoDaddy or even, you know, DOIs, for example, digital object identifiers. And that sort of authenticates where it comes from. And then we've got the aggregator, which mirrors any of these that are built. And so that's sort of another check so that we can, you can authenticate against that. And so you know that this says that it came from such and such, and this is the security on it. And all of that seems to match what's on the aggregator. So we have some of that in place. Yeah, I think it's a pretty good system, and the framework for that is very nearly deployed.
Thank you.
Sure. Thank you.
All right. Well, thank you, Jonathan, for this presentation and for what you've shared. Again, we're going to have a recording of this session where we'll make it available to everybody that participated or that wanted to participate. And we'll try and hold again another webinar, Jonathan, whenever your schedule permits, with some of the people that couldn't make it during this later time. And also try to work independently with the different groups that we have to start submitting some of these biocompute projects or objects and try it out and see how it works. So thank you everyone that joined, and we hope to see this once again, maybe in more detail on how to practically do this next time. Thank you, Jonathan.
Yeah, thank you very much. Happy to do it again sometime.
All right, great.